as we bring in our next guest, Ramshid Arjomand. He is the FSHD Society Chief Science Officer, the man. You know, I'm glad you're here, Jamshid, because you know what? I am getting tongue-tied yet again, and another guest comes to save the day to take some of my voice and help me out. Uh, and tell us a little bit about your role as Chief Science Officer at the FSHD Society. See how I had to slow that down just to get
I
it
think
out.
you need to blame Beth and Lee for scheduling somebody with a name like mine at the end of the
Thank
day.
you. Thank you for saying that. I wanted to say that. I didn't want to be mean. You have a great name. There's nothing wrong with your name. My last name is Hollenbach, for God's sake. That's not exactly short. So I get it. I get it.
Thank you. No, thank you very much for doing this. This has just been an amazing marathon you're on, and happy to help out even in the few minutes that I'm on. Yeah, my responsibility, we're a small organization, and I think what's been really wonderful and an honor is I get to work with a lot of very smart scientists and clinicians, and get their input, as well as industry leaders, on trying to determine what are those hurdles that um, they see might kind of slow their therapeutic development. And we develop plans and strategies to overcome those. And it's really a group effort. So when we put a strategy, we shop it around. We have committees of scientists, both in industry and academia, that we kind of present those ideas, and we get their feedback, and then we make sure that whatever we're developing is going to help out the drug developers meet their goals. So it's really kind of a team effort, and it's been really an amazing, um, really amazing job to have. So I'm delighted to be doing this every day.
It's funny when you said, "Oh, you talk to a lot of smart and intelligent scientists," and I've been way over my skis today. On a lot of these interviews where, and then I saw your name and I'm like, oh, jeepers, this is the Chief Science Officer. Oh man, okay, how am I going to ask this gentleman questions
So,
to?
so
to
the
get
best thing, the best thing really about being at a nonprofit is I'm not competing with anyone. So I can actually go to academics who may be competing with other academics for publication or grant money, and I can ask them questions. I can shop around my ideas and see, like, does this make sense? And I think most of the time hear an honest answer. The same thing with pharmaceutical companies. You can go and talk to them about what we're planning to do, and they'll give us their feedback. Well, if you add this, that would really be helpful. So I think in that case, it's really great. And then, with most of the academic scientists or clinicians, since we do offer grants and we do support them, I can ask really dumb questions and they'll never tell me that I'm dumb.
Oh, good. Crazy, right? 2020. I brought this up. I mean, everybody knows elephant in the room with COVID-19, but despite all of it, what were some of maybe the high points of this past year?
Yeah, the great question. I think when COVID started at the beginning, we didn't quite know how this was going to affect research, and we had a couple of major meetings planned. So we had the International Research Congress, we had the Voice of the Patient meeting, we had Patient Connect, and all of these were supposed to take place in DC. And we were quite excited. Election year, for international meeting, people coming to DC, that was going to be quite exciting. We had a great venue picked, and we kept watching it, and things just were getting worse and worse. And so at some point we thought, all right, we're going to have to transition to a virtual format, and no one had done this, right? We were just learning how to use Zoom and Teams. So to actually run a meeting, an international meeting, was just seemed really difficult. And I think we just as a group, like, and this is, I really have to give credit to everybody on not only at the society, but all of the people that were on our organizing committees.
Mm.
They did an amazing job. They actually told us, I thought at the beginning, I was thinking we're going to have an abbreviated meeting. Maybe instead of having all the sessions, we're going to have maybe an hour or 2 hours.
Yeah.
And the researchers who were on our committee told us, no, if you build it, they will come. People have already allocated 2 full days and all the travel around it to come to DC to attend this meeting. So if you have a full program, people will come
Mm.
and they'll attend. And they were right. Actually, by having a virtual meeting, we were able to reach a lot more researchers. We had about 100, typically we have slightly under 200 people attend the meeting in person. We
Yeah.
had close to 300, I think 280 people
Wow.
came. And we had people from all over the globe, people from Moscow University, all over people from New Zealand and Australia, Japan. It was absolutely amazing. And then what we were able to do also was we pre-recorded all of our speakers, and then we had them live for Q&A. And
Oh.
so that took a lot of the pressure off from people having to connect and stick to a rigid time,
Mhm.
because they had already pre-recorded their talk. And as their talk was airing, they could see the questions that the audience member were asking, and it was just a very smooth transition. So it was a great success. And then the voice of the patient meeting was just incredible. That was, I had absolutely nothing to do with that. uh, my colleagues did all of that, but it was just spectacular.
Oh.
And the report that the summary report that's just been submitted to the FDA was really fantastic. And now we're actually trying to build on that, trying to see how can we integrate the voice of the patient report and outcome measures. And so we're speaking to people who have done this and trying to learn from them. And that was some of our activities for 2021.
Wow. So are we hoping that we can do kind of more of the regular scheduled program for 2021? Is that the
That's
hope?
what we're hoping.
Wow.
We're planning for an in-person meeting for the IRC in 2021. And actually, starting the year that I joined in 2019, we actually took that meeting to Europe. So we were now going to alternate every year between the US and somewhere in Europe, and really truly be an international meeting. So we have it scheduled in Leiden in the Netherlands. Hopefully we can meet in person.
Mhm.
If not, I think we're looking to really build on the experience that we've gained in 2020 and make it even bigger and greater. So there were some good lessons. We realized that even if we were having the videos available for 24 hours after they had aired, that wasn't enough for people who are calling in from Asia or Australia. They needed a little bit longer time, so we would keep those videos up longer and things like that.
Yeah.
Yeah.
Yeah. It was interesting. I'd imagine to be in the room when you all are figuring out how do we adapt these meetings and quickly pivot and change and educate yourself on how to do it. Crazy times. I think
Absolutely.
become master adapters throughout this year.
Yeah, so
Uh,
we were lucky to really have great people, so consultants, and we reached out to really great video companies that helped us with all of that and kind of walked us through what the process would look like. So so that was great.
there's always like this like North Star pointed in my mind of this, treatments by 2025,
Hmm.
right? Mark Stone, I'm going to blame him. I'm throwing him under the bus. He's the one that he's the one that said it. No, I love it. I love the fact that he put it out there. But with the COVID-19 hiccup, does that put a little delay possibly in that goal?
So um, I don't think so. I think so for instance, for 2020, we had planned for several workshops to tackle certain initiatives that we had identified in 2019. And actually, I think Lexi Pappas earlier mentioned that she gave kind of a solo testimony in front of the FDA, NIH, and industry collaborators earlier in the day. I think she was mentioning that. That was the meeting in DC. We had actually invited all of the pharmaceutical companies, biotech companies, and all of the a lot of key academicians and clinicians, as well as the regulatory agencies, so FDA, NIH. They were all in the room, and we were trying to ask them, if you're developing a drug for FSHD, what are some of the hurdles? What are some of the gaps in clinical trial readiness? And this is kind of the initiatives that we're working on trying to resolve. So we identified several of those. There were 4 in particular we were going to tackle in 2020, and we thought we would have a workshop around each one of them, 1 per quarter, and invite key opinion leaders, key academics, experts, as well as maybe some companies that specialize in whatever the topic is, to come, and we can brainstorm and come up with a plan to take forward. And then we would have kind of a consortium of a collaborative consortium with pharmaceutical companies where they would help fund these activities. Well, because of COVID, we couldn't really convene this. So we had one that was a virtual meeting, and it actually worked out great. We had about 40 participants, which was a lot more than I thought would join. And people had great ideas, and it worked fairly well. And then we didn't get that comprehensive plan, but we got some really good experiments that we could carry out during COVID. So there were simple experiments, we could outsource a lot of it, and we put those in place, and now the results are coming in, and they actually look very promising. So these are all tools that's going to help other drug developers in like assays and things like that. So I'm very excited that despite COVID, despite having laboratories being shut down, we were able to do a lot of little pilot experiments, and a lot of these initiatives actually moved forward. So that's very exciting. And I think we can pick this up in 2021 and then go a little bit bigger, and kind of follow up on these initial findings and develop some better assays or something that's more robust.
Mm-hmm. Wow. Well, it's good to know. I mean, yeah, it sounds like a little bump in the road, but said it straight and onward we go. That's always good to hear. When it comes to the question asked, because we are, raising money today, Giving Tuesday, and people have options, right? There's other causes that they donate to. Maybe they divide their money, maybe they don't. We're trying to get theirs. Let's just be honest to it, where I want your money for our cause, because this affects me, my family, and so many now of new and old friends that I've created through this society. Now, people are going to ask, what will the society do with the funds we are raising today?
Sure. So essentially, I always tell my colleagues at the society that we fund a lot of the research and we support a lot of researchers. So all of their efforts in raising money, I basically then try with input from committees. It's not my solo discretion. I don't allocate funding on my own. It's always in consultation with the committee and their approval, but we fund these initiatives. And so, for example, an initiative that I just mentioned, a biomarker initiative, and I think Lucienne Ronco touched on this from Fulcrum, because it's very difficult to measure DUX4 level, and they do muscle biopsies and look for things that are regulated by DUX4. Well, muscle biopsies are quite invasive. So what we were trying to do is get something that you might be able to measure in blood. And there's been other studies, but most of these studies, the way they are set up is they're discovery studies. You take blood from 20 patients and 20 unaffected people, and then you run these discovery studies to see what's different between them, and then you try to replicate the finding to see, is it robust enough? Does it stand up to a retest? And a lot of the initial publications, they don't reproduce. We took a different approach, and this is again in consultation with this virtual meeting that we had. We know DUX4 activates a bunch of genes, and so we looked at that long list of genes that get turned on and asked ourselves which ones might be secreted in the blood. If we might get lucky, something might get secreted in the blood, and then we took one other step and we looked at which ones are there are there assays for any of them that we could determine? So there are a lot of assays that have already been developed for immune markers and things like that. So we were trying to create these Venn diagrams, and then we identified 3 or 4 possible candidates, and we ran a very simple experiment. And so these were the pilot studies that the society funds, and we got interesting positive results. So now we can go to the next step. So one of the things that I'm very excited about would be to go to the next step. We have to not only repeat that experiment, and these are all done with very simple models, cells grown in a dish. So if it replicates there, then the next step is to run the same test but using patient samples and see if it validates. So all of the kind of the funding that's being raised today, those can go towards these initiatives that will help every drug developer in the field, because they can all use assays that measures DUX4 activity. It helps them with dosing. It helps them determine if the drug is really working. So that would be a really useful tool for everybody.
Wow. So yeah, I mean, this money is going to this key research. I mean, it is needed in a large way to help fund what all this is, all the inner workings. I mean, it costs money. It costs money to get.
Absolutely. But it was so during the pandemic, I have to give credit. There's a researcher in Colorado, Sujatha Jagannathan, who graciously cultured the cells for us, and Amy Campbell, who's in her lab, did the experiments, and then the cost really was in this company that had developed the assays. We just sent them the samples, and they ran the assays and gave us the results. So a very, very quick and relatively inexpensive. Obviously, the next set of experiments are going to be a little bit more complicated. This was a quick go, no go experiment. The next ones are going to be a little bit more elaborate, and so if they replicate there, then we can go to the really interesting ones, which are the patient samples. And so, yeah, we need to kind of build on that. And then to once we have enough results that are compelling, then we need to go really big and do a massive study. And this is where we're going to try to get industry to help co-fund the bigger clinical studies.
Mm-hmm. Still more work to do then. I mean, we get the donations today. We got to keep at it.
Yeah, I've been running high on these good results for quite a while. I think we got the results about right before Thanksgiving. And as a scientist, I don't know if you've spoken to a lot of scientists, but a lot of the experiments we run don't work. And so right around the time when you're about to throw in your hat and quit, you get this glimmer of hope, an experiment that works and gives you really cool results. And so that totally keeps you motivated to keep going with it. So I'm running on those great results right now and very excited about it.
I share your excitement. If you're excited, Jamshid, I'm excited because you're a smarter man than me, and the way you've interpreted it makes me excited because this makes sense. And I appreciate you coming on today to kind of go over what all the things you're doing for the FSHD Society. Thanks for being here. I appreciate it.
Oh, my pleasure. Thank you so much for doing this as well. And thank you to all the participants and the listeners and the people who are donating. This has been really wonderful. Thank you to everyone.
Yeah. Thank you. Have a good night. Thanks for your time, sir.